Okay, so we're back. So let's get going with the handout on this. Um, so ultrastructure and ultrafiltration. So let's talk about that and kind of let's recall what we learned. Okay, so this is the part where the blood comes into the glomerulus. You have um, the blood is under high pressure, and so in this case, blood will leave the glomerulus in a process called ultrafiltration and going into the Bowman capsule. At that point, it will be called filtrate, okay, and that filtrate will contain these stuff, okay. Now, let's go into a little bit more detail. So in this case, you have the afferent arteriole and you have the efferent arteriole. Now, I'm looking at the diagram. Now, even though the position is correct, I think we kind of talk about the blood flow slightly differently in our class. So in this case, the structure of the efferent and afferent and efferent arteriole, it is different. Okay, it is much wider in this one, the incoming one, the afferent one, than the efferent one that is narrow. So imagine like a 4-lane highway worth of blood going into this glomerulus area. But at the end of it, you only have a 1-lane road exiting. Okay, 4-lane highway going in, 1 lane going out. So as you can imagine, you'll have a traffic jam because this will be bottlenecked. All that blood that goes in will be bottlenecked. So it will be slower leaving here, but at the same time, the blood in here will be high pressure. It's like a traffic jam. So at that point, this high pressure will cause the blood and the blood vessel. So if you look at it, this is the blood vessel, and then you have the fenestration spot, you have the basement membrane, you have the podocytes. So these slits here, these are the fenestration. Okay, that will go leave. It will push. So it will push the blood, leaving these fenestration here. And we talk about fenestration. The fenestration here. In before the break, I kind of mentioned something about the castles in Europe, and the walls of it have little, very narrow slits which allow people to shoot arrows through. So in this case, those are fenestrations. So if you look here, that's another diagram of looking at it. You have the blood vessel here. So the blood will go through these fenestration slits, past the basement membrane, go past the podocytes, and then becomes the filtrate. Now here, this part too. So it's not just the fenestration, but also the basement membrane, the podocytes. All this here prevents the large molecule from going through. So it's not just the fenestration. You actually have quite a bit of other stuff going through. And if you think about how much filtrate goes through, it's 1 liter every 10 minutes. This is really fast. Okay, it is a pretty fast flow rate. So it is high pressure. I don't know if you hear the baby crying in the next room. Hold on. I'll pause. Okay, I'm back again. Fun. Working from home. So anyways, now, so where we are. So ultrafiltration. Blood goes through the fenestration into the filtrate. That's where we are at. We talk about these structures, basement membrane, podocytes, all these, in addition to the fenestration, prevent large molecule going through. Right. Now we're at the proximal convoluted tubules. Okay. So a lot of it is reabsorbed by active transport. Now, one thing which I don't know if you remember this from your digestion section previously. In probably in grade 10, I think you guys learned a little bit about digestive system. Now, this part here, you will see these features again later on when we learn much more about the digestive system, because these are basically features not just for the proximal convoluted tubules, but for a lot of our system, so that we can actually quickly absorb material through a membrane or through a wall or something like that. This, you'll see this again in the small intestine section. So wall is only one cell thick, so the material substance doesn't have to go through a long journey, so the distance is shorter. Interior of the tube is called lumen, where the filtrate go through. Now, I don't like this diagram that much because you don't really see it that well. I like the diagram in the study guide. So if you look at the study guide, selective reabsorption, this is the proximal convoluted tubule. Okay, proximal convoluted tubules. Here's the lumen. Okay, lumen, that's the hole in the middle. That's where all the filtrate's flowing through. So as you can see right now, you have lots of mitochondria. No, not mitochondria. Lots of this microvilli. This increases the surface area. Basement membrane, that's just basically the membrane of it. But all this is only one cell thick. So the material from the proximal convoluted tubule, from the filtrate, goes from here to here, only go through one cell. And you have a lot of mitochondria here because you need mitochondria for ATP, because ATP is used for active transport. Okay? So all these features, wall, one cell thick, anterior wall, lumen, microtubule, microvilli, these features, I'm going to go through here. You're seeing this here in the proximal convoluted tubule in the kidney. You will see it again in the small intestine. Now, when I compare the notes here versus comparing the notes here, what I do find is that this is actually quite, I think it's a lot more material than you need to know. So we're going to I'm going to cover this looking at here. Okay. So if you look through this, you're talking about large surface area, you're talking about the pumps, using ATP to pump. All the glucose of the filtrate are reabsorbed. So all the glucose reabsorbed. 80% of the minerals, sodium, are absorbed. Active transport solutes make the total solute concentration higher in the cells than in the filtrate of the tubules. Water therefore moves by osmosis from filtrate to the cells in the adjacent hood. Blood. So blood cells are around here. It's not shown in the diagram. Okay, are not shown in the diagram. In this case, basically you're using active transport to pump the solute. So when we're talking about the solute, we're talking about the sodium, we're talking about the sugar, amino acids, all these stuff. So those are pumped out by active transport. And because of that, in here, this will be a lot more saltier, or a lot more solute, higher solute concentration. This will be lower solute concentration because of the active transport. And because of that, therefore, the water will move by osmosis from the filtrate into the cell and then on to the adjacent blood vessels. So here you have capillaries on the other side, which is outside of it. So that's how the proximal convoluted tubule works, and selective reabsorption. Okay, keep in mind, going back to this diagram at this point, all the glucose are out, amino acids are absorbed, etc., etc. Descending loop of Henle, ascending loop of Henle. So we talked about this before. It is the structural difference. This is water permeable but not permeable to salt. On this side, the ascending side, it is permeable to salt but not permeable to water. We talk about this diagram. So let's look through this. The water will go through here. Descending impermeable to salt. This ascending one permeable to salt, impermeable to water. Number 3 at this point. So at this point here, you actually have to pump this part here, because so much water has gone out. This filtrate here is actually saltier than the surrounding membrane, so there's passive transport. But as the sodium and the chloride, the ions leave here, by this point, it is active transport, because at this point, the surrounding tissue actually is saltier than this, so you actually need active transport. So we talked about this before the holiday, okay? Pause here again. We talk a lot about stuff. How are you doing? Do you have any questions you want me to go over or talk about certain things? Now, in the video, we kind of learned that we have a distal convoluted tubules. More salt is exiting. More water is also taken out from the filtrate, and then we go to the collecting duct. Okay, let's see if I can move. Collecting duct. And we didn't really talk much about the collecting duct when we draw this diagram, but that's a part where water content. Okay, here's a collecting duct. This is the water content where is adjust. We're trying to adjust how much water we want to get rid of or not. Normally, we try to absorb a lot of the reabsorb a lot of water to the surrounding tissue. So your pee is actually more concentrated. Hello. Okay. So permeability of the duct around here, how permeable water goes through, it's under the control of ADH. It's by the pituitary gland. So it either makes a channel more porous or permeable or not. Dehydration, release ADH, which is a negative feedback. So if you're actually dehydrated, you actually have more ADH, so you reabsorb more water. Okay, so now loop of Henle, this is the part where different, I think the video talked about too, the loop of Henle, the length of it is different for different animals, and that again depends on where they live and what kind of habitat they live. The longer it is, the longer it is usually means more water is reabsorbed. Usually you're talking about a desert animal. The composition of blood in renal artery is different from renal vein. Okay, so at this point in the renal artery, you have more excess water, more excess salt, toxins and other things not metabolized. So that's why if you had beets and about a few hours later you want to go pee, you will notice that your pee looks red. It's because those are the substances that are not fully metabolized. Okay, so the composition of blood is different. This is simple. What's the difference between dehydration and overhydration? So if you drink a lot of water, okay, what will happen to your blood? Now keep in mind, overhydration is just as dangerous as dehydration. However, you do have to drink a lot of blood. Drink a lot of blood. You have to drink a lot of water in a very short amount of time to become overhydrated. So you do want to make sure that it's harder to be done. Okay, generally people get dehydrated rather than overhydrated. Okay, um, treatment of kidney failure by dialysis. You guys can read this. Um, it takes a couple hours if you have to do dialysis to go through. Yeah, a lot of actually right now with the coronavirus and whatnot, some of the people that have from this one of the stats that I've read, um, a lot of people who passed away. Okay, I'm doing something right now. Sorry. So a number of a significant percentage of people who died from the coronavirus. One of it is because they have pre-existing condition with diabetes, and so if you think about it, it does. If you have to do dialysis, you do have to go to hospital or clinic a lot more. You are more exposed to it, and that complication, it's so you get the disease because you went to have to go through dialysis and get exposed, or the diabetes with the symptoms of coronavirus. That is, I don't know. It's something worth looking into. Um, but basically, if your kidney cannot filter out the blood itself, you'll have to go through dialysis unless you get a transplant. Okay, last but not least. So what happened if you go for a P test? What happened if you found various things? You can actually find drugs, yes, but that's one way to see if you can find drugs. Usually those are relatively short term, like recent stuff. But again, I'm not an expert in it. I only heard it from an uncle who worked in the police force before. So, yeah. So in this case, I don't know. I don't know what's the stat. If you're really curious, go look it up. But that's what I heard. But if you have glucose and protein, you have diabetes. If you have protein, kidney damage. If you have white blood cells, you probably have a UTI. Red blood cells, kidney stone, or tumor. Okay, that sounds serious. So, pause again. You have any question? Now, what I would ask you to do is to finish this you try question. There are a lot of things that these are a lot of this. When I look through this, a lot of these are actually good practice questions for your test. Again, I have no idea when the test will be, simply because I don't even know how the schedule might look like next week or the week after. So in this case, I'll let you give you more information as we go, but that's what I'm wrapping. So we're done with the kidneys. We're gonna see what we're gonna do next. Okay, bye now.